Thanks to everyone for joining us today for the Spiny Surveillance webinar. This is our kind of spring training and kickoff. The real season doesn't start till later, but we want to make sure that you're prepared and have time to get equipment and all the things that you need if you want to participate in Spiny Surveillance this year. Lots of this will look familiar if you have done this in previous years. There's a couple of updates that you'll see along the way, mostly to the app, so it'll be kind of towards the end. But I am joined here by Don Brandstratter and Val Brady, who were both part of the research crew that developed this program. So they're going to kick things off to talk about some of the background about spiny water flea and where the program came from and why the protocols are the way they are. And then I'll join in and chat a little bit about how to submit your data and what to expect in terms of like communication and things like that moving forward. And then we'll have time for question and answer as well. So Don, I'm going to turn it over to you.
Okay, very good. Thanks, Megan. Welcome, everybody. Thanks for being here. As Megan noted, we're going to work through this bank of slides, sort of backfill the background on how this project originated, and then take a look at some particulars in terms of how you can get involved. So this is spiny water flea surveillance. The program was developed by MAISRC and people within MAISRC, and it engages volunteers and local managers statewide in Minnesota to help out with surveillance. And just as a visual, the person in the slide to the right there, that's what surveillance looks like with our approach, taking these nets and lowering them down into the water, bringing them up. And the target of interest, spiny water flea, is shown on a fingertip there in the lower left. So go ahead and advance, Megan. Or do I control this, Megan, or are you? Or yep, there we go. Thanks. Okay. Okay. So what is surveillance monitoring? This is really just strategically intentional coordinated surveys to help update distribution data and identify new populations early on. And so we do surveillance monitoring for a whole variety of different organisms across the landscape, invasive, non-invasive, looking at changes in range distribution. Next slide. So spiny water flea surveillance in particular, surveillance monitoring in particular. So this program was originated in 2022, and the intention was to, as it's noted there, equip volunteers and local managers with the knowledge and understanding, as well as the gear to do surveillance for, and then to report potential positive hits or finds of spiny water flea. And the surveillance monitoring became another branch, if you will, of this broader umbrella program within MAISRC called Stop Spiny Water Fleas, Stop the Spread. And so there's a whole variety of different resources and materials that you can access through this website, one of which is gaining a better understanding and directives for how to do the surveillance monitoring. Okay, next one. These are the people that were behind this. The founders include myself on the left there, Val Brady in the middle, who's with us today, and then Meg Durr, who spent a number of years at MAISRC and has now moved on. A couple of the founders, the two of us, are still here working on this, but really the legwork for this in the last couple of years has been advanced and carried by Megan Weber, who's with us today, and then Michelle Isaacson. And so we're sort of passing the baton or passing the torch to them. But a number of people have been involved in the development of this program. So why conduct surveillance for spiny water fleas? I mean, what's the point here? What's our mission? Well, to date, and when I say that, I mean 2025, we have about 70 or so Minnesota water bodies that have been reported infested. That's probably an exaggeration based on the way the DNR reports infestations. If there's an infestation in one lake, a connected water body will also be deemed infested as well, even if there isn't strong evidence of spiny water flea being there. So in reality, there's probably only 3, maybe 4 dozen lakes that actually have confirmed infestations. And there's been very few since 2016. Only a few lakes, Snowbank Lake and maybe Seagull Lake and another one up on the northern tier of Minnesota have been reported infested. We know that there are many, many more lakes based on basic research that are vulnerable based on water quality, the size of the lake, the depth of the lake. And so there's clear evidence that thousands of additional lakes in Minnesota could probably harbor a population if they were to arrive there. And on top of that, there's nobody responsible. Nobody is being paid for or supported to do monitoring on lakes across Minnesota. So it's really at the hands of this surveillance volunteer monitoring program to continue to evaluate lakes and for presence of spiny water flea. Okay, next one. Okay, so this is sort of continued. Why conduct surveillance for spiny water flea? Well, they spread. We know that they can spread quite readily, and we think, and we have evidence that recreational anglers and boaters are probably a prime pathway by which they spread. We know that some of it, so there's not a lot of lakes that are infested with spiny water flea at this time, but the ones that are some of our premier, largest, most recreationally attractive and active lakes in the state. Lakes like Lake Mille Lacs and Vermilion, right? And Rainy and so forth. And so these huge water bodies, despite it just being one lake, it has the potential to engage a lot of recreational activity coming to the lake, leaving the lake. And so the opportunities for a spread from these, what you might call hub lakes, is really high because of the recreational usage rates of these lakes. In addition, spiny water flea, when they're clumped up on lines like you see there in both of those images, they're relatively easy to detect. But as individuals, you know, they're essentially microscopic. They're about total length, the length of your maybe your index finger, and they tend to be relatively transparent as well. Okay, next slide. Okay, so they just sort of continued here. We're worried, and we have every right to be worried about new infestations because despite the small size of the organism, they can grow rapidly in number, and they can impose really big changes to food webs, and that can cascade to changes in water quality as well. Once they're in a water body and they're growing readily, there's no known means at this time to eradicate them. And so the only decent way to control this organism on the landscape is just to prevent its further spread. With knowledge about where spiny water flea are in Minnesota, that's going to help us focus prevention and outreach efforts. So that's one main reason why we should do surveillance. Okay, next slide, Megan. So just a repeat of that statement, early identification allows us to focus prevention and outreach to stop further spread of spiny water flea. In addition, early ID, so the types of information that would derive directly from you and the surveillance volunteer monitoring, that information could allow us to determine potentially the source of where that spiny water flea came from, and ultimately help strengthen prevention efforts. In addition, early IDs, and I underscored that there are no new identifications. That's critical information. It allows us to understand patterns of regional dispersal, timelines, and spatial patterns of spread in Minnesota. For example, if we start to see a whole series of new confirmed infestations, that's all really important information. And we've been running this thing now for a couple of years, and we've had a lot of effort, a lot of boots on the ground, and we haven't had a single new confirmed identification. And so we'd have what you'd call zeros, right? But in this business of looking at range expansion and spatial patterns, zeros are really critical information. That's knowledge about what's there, what's not there. Okay, not doing this work at all, we're sort of blindly going forward with respect to distribution of resources and an understanding of where spiny waterflea are and where they aren't. I'll just say currently right now, it's honestly sort of been a little bit surprising, I think, to scientists and lake managers that we have no confirmed infestations over in the Brainerd Lakes area, right? Like Cass Lake and some of those Winnibigoshish Lake and Leech Lake, for example, and all those lakes over there are still, as far as we know, spiny waterflea free, and that's good. Okay, next slide. Okay, this was just sort of something I put in here for fun, is to sort of think about an invasive species and how we learn about what the impacts are of an invasive species. And, we know a lot about how food webs, for example, of lakes work and how native species interact. And sort of an interesting, perhaps, analogy to that is to sort of think about a chess game. If you've played chess, you understand the role of the king and the queen and the bishop and the pawn, and you sort of know how these pieces interact, and you can think about maybe those interactions as competitive interactions, sort of predator-prey, consumer-resource type interactions. And so what scientists and managers were struck with when spiny waterflea began to arrive on the landscape 20, 30 or so years ago was like a new chess piece in the game. And we really had no idea what its role was in the ecosystem, in the food web, how it was going to interact with the different known species that were out there. And only through experimentation in the lab, as well as new infestations in ecosystems and following those new infestations and looking at the outcomes of that, did we, and we have begun to really understand quite confidently what impacts and roles spiny waterflea plays in ecosystems. And while some of this has been done in the lab, the real gold standard has been tracking how natural food webs change from before an infestation to after an infestation. And so by experience and sort of observation and inquiry, we've come to understand what the role of spiny waterflea now is in a food web, and it's not good. Okay, next slide. So one way to sort of to think about that, the way in which energy and nutrients flow in a typical food chain or a food web in an uninvaded lake is from the primary producers in the upper left, small microscopic plants called phytoplankton or algae, into small microscopic animals called zooplankton, which are fed on by all fish when fish are young, and even some adult fish stay zooplanktivorous. Fish like bluegill there and perch might continue to eat zooplankton throughout their adulthood. And then those provide the energy source for bigger piscivorous sport and game fish like walleye and northern pike. And so this is what an uninvaded food web looks like. So when we add spiny waterflea to a food web, be the next slide, they sort of engage this flow of energy at the zooplankton level. And so spiny waterflea they're carnivores, they're not herbivores, and they eat zooplankton. And so they basically compete with the zooplanktivorous fish for the same common food resource. And because of the numbers of spiny waterflea and how fast they grow, they can suppress natural zooplankton abundances significantly, and they do. And we see this time and time again. So in the Lakes of Voyageurs National Park, they experienced about a 50% on average decline in zooplankton abundance throughout the growing season after spiny waterflea arrived in that lake. And part of that destruction to the zooplankton is direct consumption. And it's also the case that when spiny waterflea are around, a lot of zooplankton will spend more time in deeper depths of the lake where it's colder, and they themselves will grow even slower. And so it's not only that they're getting eaten, but they're just growing slower. And all that means less food for zooplanktivorous fish, which ultimately means less food for walleye and other what we call piscivorous fish, and evidence from Gretchen Hansen and other folks has shown that growth rates of yellow perch and even walleye now in Bythotrephes infested lakes is less than what it was prior to invasions. Okay, next slide. Okay, so why can duck spiny water flea continued? We're really making the case here. It's easy stuff. It's fun. You get to get out in the field and you get to do the, use the same sorts of equipment and engage the ecosystem in the same way that we train scientists do. This is how we do it as well. There's no laboratory test needed. The number of items that you need, there's a couple that are specialized that we'll talk about here that are important, are critical. But again, the research-based guidelines can greatly enhance efficacy of survey methods. And so there has been some research that's been done to ask the question, how thorough does a survey have to be in order to be effective at determining sort of a yes, no, a presence, absence, whether spiny water flea is in my lake or whether it's not in my lake. And we've engaged some of that research to develop the protocol that we're providing here for the for you in this monitoring effort. Okay, next slide. Here's just a couple of things to think about with respect to this, the timing of your monitoring efforts. We'd really, we're best off, you're best off, best case to detect spiny water flea is during the open water season from June to October. And that's why we're running this webinar here in early May. We still have a month or so before spiny water flea in its active free swimming stage really begins to show up in the water column. And they do. So at this time of year, essentially throughout the winter and even now in May, all of the spiny water flea that are in any infested lakes are down in the sediments in this resting stage, this diapause state of no growth, and they're essentially undetectable. And about now, about a month after lake ice off goes off lakes, those eggs get a cue from light and temperature, and they hatch, and they give rise to female spiny water flea, and like you can see in the upper left there. And those females reproduce asexually, clonally, all summer long. And over the course of 7 to 10 days, they can produce another crop of offspring, 5, 6, 7 offspring. And each one of those only takes a week or so to grow up and produce offspring. So they can really rapidly populate a lake, and they'll continue to reproduce asexually like that all summer long, all the way into October. And that by far is the best time to detect spiny water flea in the lake. Come fall, when food declines and food concentrations decline and temperatures deteriorate, they return back to this diapause resting egg stage, which settles down into the sediments. Nothing that you're seeing here on this slide is life size. The spiny water flea in the upper left, again, they reach about maybe a third to a half inch in total length, and those eggs down in the lower right, those two brown circles, those are the resting eggs. Each one of those is about a half a millimeter in total size, and so those are definitely microscopic. So I'd like you to try to focus your efforts for detection. Best chance detection is around August, September. It's when densities are maximal in lakes. Okay, another thing to think about when you're doing your surveys, and so the next slide, Megan, is that, okay, so we're gonna have you collect with zooplankton nets, and they can certainly be used during the day. There's nothing wrong with that. If there's a lot of spiny water flea in your lake, you'll have a reasonable chance of detecting them during the day. But very early morning and late evening, even after dark, if you have the capacity and you feel it's safe enough to go out after dark and to survey, that will we really maximize your chances of capturing spiny water flea if they're there. And that's because, like other zooplankton, spiny water flea move up and down vertically in the water column over a day-night cycle. So if you go to the next slide, Megan. And we know this from research in a variety of different lakes, including these data came from a lake just north of Duluth called Island Lake Reservoir. And we used that box trap shown in the upper right there and lowered that to successfully in the water column and collected spiny water flea at different times throughout the day in 3 different months. And so each of those panels shows these bars that go from left to right. And the longer the bar from left to right, that's just indicating how many spiny water flea we detected at that depth. And then the top of that panel is the surface of the lake. And then the bottom of that panel, that 12, means that was 12 meters depth in the lake. And then as you kind of go across those panels from left to right, there are 6 panels, and you can see the sun in the first 3. Those were during the daytime, 10 in the morning, 2 in the afternoon, 1800 hours at 6 in the evening. And then nightfall, the moon comes out, and you can see those bars all grow in size. That's just evidence that spiny water flea have risen up into the water column. It's also the case that they might actually be avoiding nets and traps during the daytime. They do have this big eye, and they're known to be visual predators. And so it's possible that they're visually avoiding the net as well. So it doesn't mean, as these data show here, that you can't catch them during the day, but you're going to amplify your chances if they're in the lake, if you can go out in the early morning, the late evening, or even after dark. Okay, next slide, Megan. And then the other thing that we know based on a lot of research is that during the growing season, so as these data indicate here, from July to late October. So what we're looking at here, I should set this up, is month on the lower horizontal x-axis, and this is back in 2002. So May through November, and then the vertical axis there is density. So this is numbers of spiny water flea that we estimated in each cubic meter of water. You sort of think about those numbers, right? 50, 100 animals per cubic meter of water. Think about what that translates into in terms of total number of animals in a lake. And it's just millions. It's millions and millions. But what you can see there, and the point of this is to see that the densities at a few different locations in this lake, they go up and they come down, and they go up and they come down. They kind of wax and wane. And this is fairly typical of natural populations to go through these oscillations or these cycles. And they're just tracking abundances of their own prey as they reproduce heavily, and then they sort of have a die-off, and then they reemerge again back and forth. And so the point here when we're doing surveillance monitoring is that if you go out and you don't detect any spiny water flea, it may just happen that you were out sampling at a low point in the seasonal cycle. And if you wait a week or 2 or 3 and go out again, you might hit a high point. And so if you can, and when these peaks and valleys occur, is anyone's guess. And we've studied the same lake 3 years in a row, and these peaks and valleys happen every year, but the peak and the valley, the timing of that is different. It's driven by a lot of factors, temperature and food and whatnot. But the point is, if you could go out 2 or 3 times in the same lake, that will be better than just going out once in that lake. But if you can only go out once, that's fantastic. Okay, we don't want to minimize any effort that you're putting forth. But if you can go out a couple, 3 times, that could again amplify your chances of detecting them if they're there and you didn't detect them in the first time around. Okay, next slide. All right, so that is the background, and once we get to Q&A, we can certainly address any questions that you might have had there. We're now going to sort of transition a little bit into the effort, the boots on the ground. What is the equipment that's needed? How do I actually go about doing this? So here we have a list of equipment needs. The most likely thing that you don't have in hand, although you might if you've been doing some zebra mussel monitoring, for example, is a zooplankton net attached to a rope. And while any net, any plankton net, whether it's for mussel veligers or zooplankton, will work. What we call a 500 micron net is ideal. And when we say 500 micron net, that just means the holes or the apertures are each half a millimeter or 500 microns in diameter. And the only reason why that net is ideal is because a lot of the stuff that will clog the net and that will make your collection really thick and challenging to go through is less than 500 microns in size. So things like phytoplankton or algae and small zooplankton, which are by far the most numerous particles that are out there in the water, all that stuff, that microscopic, super microscopic stuff, will go right through the 500 micron net, and you'll be left only with spiny water flea if they're there, and some of the other big zooplankton, and that can help you be more efficient at detecting whether or not spiny water flea are there. So a net is important. A white plastic dish pan or similar shallow tray, a pair of tweezers, an instrument, something like a fork that you can poke around and sort of delicately move plankton here and there to try to isolate something that might look like a spiny water flea. A squirt bottle or anything to rinse off your net. It'd be nice if you had a camera. A smartphone is perfect. Flashlight or a headlamp. A magnifying glass that you can use to obviously enhance a live animal to get a better sense of whether it matches or not the image of a spiny water flea. Something to write on. Write in the rain paper is good stuff. And then something, if you have a specimen that you're pretty confident is a confirmed spiny water flea, based on your best effort, if you have a jar, some small clear jar or vial with a sealable cap to it and some rubbing alcohol or isopropyl alcohol that you can add to that, 70% is good, that will preserve the specimen long enough for somebody at the DNR or one of us to confirm the identity. That would be most ideal. Then we have all the evidence that we need for a positive identification. Okay, Megan. Yeah, and this is what some of that stuff looks like. So 70% rubbing alcohol there, dish pan, even a white bucket, for example, sort of a shallow rim white bucket, a squirt bottle of any type, even if it's like a biking water bottle, something like that, anything that you can use to rinse down the net in your equipment, and then something to write on as well. All right, so choosing a plankton net, Meg Durr did a lot of legwork, and we've continued to go back to this company, Aquatic Research Instruments. They're well aware of our citizen surveillance monitoring program, and a number of our volunteers have reached out to them and purchased the net. They're well aware of the net that we're suggesting be purchased. So it's a company out of Idaho. There are other companies that will also sell nets. Again, the ideal net has an opening, a mouth opening. So you can see that steel ring on top. The ideal net has an opening that's a half a meter in diameter, and it's outfitted with 500 micron mesh. But if you have a net that is a third of a meter opening and is outfitted with 100 micron mesh, use that. If there's a lot of spiny water flea in your lake, it'll certainly work. So we have more information, or we can provide more information about companies where a net could be ordered and sourced from. Okay, Megan. Oh yes, a number of years ago, we put together some fun videos. I went out into the field with an undergraduate student who filmed various stages of what it would look like to to go through the process of sample collection and identification and preservation. So maybe we'll play a minute of this. You can get a sense for what these videos are. I think there's 4 of them in total.
Hi everybody, my name is Don Brandstrader, and I'm going to show you today how to use a plankton net to collect Bithytrephes, otherwise known as spiny water flea. So we're out here on Island Lake Reservoir in Duluth, Minnesota, and this is a lake that's known to have abundant population of spiny water flea. It is a relatively windy day, and so we anchored, and that's really important to anchor if there's really any wind whatsoever, to make sure your plankton net gets all the way to the bottom of the lake. We're in a location that's about 13 meters deep, and so I'm going to deploy this net to 12 meters and bring it to the surface. This is the net itself. It has a steel bridle, and it has an opening or a mouth diameter of about 1/2 meter, and the entire net is 1.5 meters long, and it is outfitted with Nitex mesh that has holes or apertures that are 500 microns in diameter. This is sort of an ideal net for collecting spiny water flea, but other variations on this theme will work as well. Down here at this end is the collecting bucket, sometimes called the cod end. And one useful thing that is important is to have some weight associated with that. So here are some 2-ounce fishing weights that we coupled to the cod end with a zip tie, and that will help allow the net to go down vertically in the water when you deploy it. Okay, so we're going to put it into the water now, and we're going to make sure that the collecting bucket or cod end is submerged fully and full with water so it deploys vertically. And then we're going to just sort of slowly lower the net. That'll ensure it stays vertical in the water column. Like I said, we're in about 13 meters of water. I don't want to go all the way to the bottom necessarily because I don't want to pull up a lot of sediment. You can if you know that the bottom substrate is relatively rocky and inorganic, but I want to stay off of the bottom a little bit here. Okay, so that's 10, and then that's 11, and that's 12 meters. And when I've reached my destination, the maximum depth that I want to deploy, I'll let it sit there for a couple seconds. And then I'll retrieve it. And it's just a relatively simple hand-over-hand retrieve, about 1 meter a second. You don't want to stop retrieving when you're pulling the plankton net, because you don't want any animals that are caught to sort of backwash out of there. Okay, we're at the surface now, and I'm just going to lift this net out of the water. Now, one of the things about spiny water flea is that long tail spine tends to stick and ensnare to some of the mesh. And to help encourage or discourage that from happening, once you get your net to the surface, a nice way to do a rinse is to do a dip, maybe 9/10 of the way down. You don't want to put it beneath the opening ring, and lift it back up just once. Okay, so that we've made our collection, and that's all for this video. In the next video, I'm going to dispense the contents of that into a sorting tray and show you how to identify.
Okay, well, you got a good sense for how that goes. I had an undergraduate student out there who was doing all the filming, so thanks to her. Those are fun to look at, and you can go back and tap into them. The very last video that we provided was one that was titled Lookalikes, I believe, and it was intended to help everyone, kind of clarify what spiny water flea looks like and help identify, I guess, if you will, a few other organisms that are native to Minnesota lakes that you're most likely to see. So spiny water flea as a morphologically is really distinct from anything else that we have. But when you're looking at this stuff for the first time, it can be challenging, and I understand that. The namesake feature of this organism is this long tail spine, this long caudal spine, and that represents 3/4 of the total length. It is barbed with either 1, 2, or 3 pairs of lateral barbs. The first pair is up at the base of the fleshy part, and then partway back there's a second pair of barbs, and then a third pair of barbs. The tail spine often has this little S-bend in it, this kink we call it, about halfway back, and then the tail spine can interestingly be discolored. Not in all individuals, but sometimes it can, and you can see it's kind of maroon or reddish there, and then it transitions into a lighter color. That's not photographic trickery or anything. That's just what the tail spine looks like. Has a big robust thick body to it, long arms, and a really large single eye that's on the forehead. Nothing else looks like that, but the lookalikes, if we go to the next slide here, look kind of like it, right? And so the native species that probably in some ways morphologically definitely looks most like spiny water flea because it's its closest relative is this one called Polyphemus, and that's in the upper right. And Polyphemus looks every bit like spiny water flea except for the long caudal appendage. It does not have the long caudal tail spine. It's like you took a spiny water flea and just sort of excised that long tail spine. Everything else looks like spiny water flea, that big black eye up front. Polyphemus tends to be much more near shore, hiding out in the rocks and cracks and crevices along shorelines, and so you're less likely to find them out in the lake. My guess is that the organism that most of you will encounter is this thing in the lower right. This is a really common type of freshwater zooplankton. It's actually an insect. This is the aquatic larval stage of an insect that will ultimately pupate and turn into a non-biting mosquito in its adult wing stage, in which it only lives for hours or days. It looks an awful lot like a mosquito. When it's growing up in the water, it is a really interesting organism. They reach about 1 centimeter in total length. The thing is called Chaearborus. Sometimes they're called glassworms. They're predators, just like Bythotrephes and spiny water and Polyphemus there. So Chaearborus is something you might see. It has a black eye at one end, but you can see it doesn't have a tail spine, right? It's sort of long and fleshy from tip to tip. And then the third lookalike is this organism in the upper left. There's 2 organisms actually in that image. The larger of the 2, the one that's a little bit milky and ghost-like, it's not fully stretched tip to tip. It's sort of curled up there. That's called Leptodora kindi, and in its non-preserved state, it's incredibly transparent. It's almost invisible at times, but it'd be exciting if you saw it. They're gorgeous organisms, and they as well have a length like spiny water flea, but they don't have that tail spine, and that's really the telltale feature of spiny water flea. So those are the 3 natives that approach spiny water flea, either sort of morphologically and in size, that you're likely to encounter. And we've had plenty of situations in which volunteers text either Megan or me an image and say, "Hey, what is this?" And it's fun. It's exciting to see it. And I usually say it's Chaearborus. So, okay, next slide. And again, size is a key difference as well. And so this is a really cool, simple image here that I think Meg Durr took, or maybe I took this, I don't know. So that spiny water flea right there in the middle. The organisms on the right there are Daphnia, common water fleas, and so the size, I mean, spiny water flea is just outsized when it comes to native zooplankton, except for Chiabris and Leptodora. But all the herbivores, the things that are out there that are eating the algae, spiny water flea is just so much larger than it. And another interesting thing here about this image, that female there, she's carrying on her dorsal surface there a brood pouch. And honestly, the reproductive capacity is just unbelievable. And you can see in that brood pouch, you can see 4 black dots. Maybe there's a 5th one in there. Those are embryos that are getting ready to bust out. And so the reproductive investment there in that brood pouch almost rivals the body weight of that female when they're in full reproductive mode. So you might see that as well if you have spiny water flea that are really doing well in your lake. Okay, next slide. Yeah, there's a couple of them there. I don't know what more to say with that. There's a female with, yeah, okay, that's fine. Okay, so how to document a find. I think this is, go ahead, Megan, that's fine, yeah. This is your jam, right? Yeah.
Sure. Yep. So I can take over here. So the next piece, if you've gone through and have followed all the steps that Don talked about as you're sorting through what you found in the net, if you find something that you're concerned might be a spiny water flea, we have some steps that we want to be sure that you're doing while you're out in the field to report back. You'll want to take a photo, do the best you can with the phone you have. I have one more slide that'll talk about that in a little bit more detail. Record the location that you're at, the depth you had your tow at, the time of day that you did that. And then you should preserve that specimen in a small jar with alcohol. Again, Don mentioned that. You'll write down all of that information onto a piece of paper and put it in the jar. The important point is that make sure you're using pencil, because if you are using pen and you put that into a jar with alcohol or marker, that will dissolve. So if you want it to still be legible afterwards, make sure you're using pencils so that stays in place. And then you can contact a DNR specialist for whichever region you're in. That information's on their website. And again, I'll have slides with this. And we also would like you to report that in to us as part of this program. So we want to make sure there's two forms of reporting going in, one to DNR to get the new invasive species, and then one to us to report results for the program. Okay, so I had mentioned taking photos with your phone, and this is improving year after year on how you do them, but there are differences, especially depending on how old the smartphone that you have is, on a digital zoom versus an optical zoom. So if you're using a digital zoom, which oftentimes happens if you're like using your smartphone and you kind of like are pinching and spreading with your fingers to zoom, it's not actually like changing the lens. It's just, making things bigger, but it doesn't make anything sharper that's smaller in there. So your best bet is to use, if you have a phone like in the bottom where there's more than one lens, then your best bet is to use, like, usually there's a button that says like 1x, 2x. I think mine has 1x, 2x, and 5x. If you press those buttons, it's going to switch which of those lenses it's taking the image from. And that's your best bet at getting like a really good sharp image with like a true zoom. So for something tiny like that. And you also want to double check that it's in focus. So after you snap the picture, open it up and take a look, especially with little things when you're getting lenses real close. It's easy for things to come out of focus. If it won't focus with your phone super close to it, just back off as much as you need to get a nice sharp image. And then it can always get cropped down to show what you're trying to show in that photo. I had mentioned you can find information on the website to make sure you're alerting DNR to a find. So if you go to the DNR's website, on their invasive species page, there's this contact and expert section, and you can scroll down. There's a map if you don't know which region you're in to figure out who it is that you should connect with to talk about your find and to figure out how to get that sample to them. And we also have a tool within the Minnesota Aquatic Invasive Species Research Center that can help you do some prioritizing for lake samples. So you may be joining because you're really interested in monitoring your favorite lake, and that's just fine. If you're interested in monitoring other lakes as well, this can be a tool that you could use to help make some decisions about what that might be. So the website is aisexplorer.umn.edu. And once you get in there, it has some tabs at the top, and the one that lets you sort by risk for spiny water flea is underneath the prioritization for watercraft inspections. So on this lower corner, you can see like percentage of boats to inspect. If you just bump that up to 100%, it'll kind of give you some rankings here, and it can give you some ideas about like which of these water bodies this modeling is seen to be the most risky for spiny water flea introductions. And that might be a way that you can make some decisions about what lakes you might want to monitor as well. Okay, we also have an app. So I had mentioned reporting data into MAISRC, and that's using this app. This was developed thanks to Alex Badges, who's the quantitative ecologist at MAISRC. And the app has been refreshed again, so I have some updates to share with it. But it's just going to ask for some basic information like your name. We removed the password this year too, so you may remember of like having to enter a password and it would change, and then you wouldn't be able to enter your data. You shouldn't have to do that this time around. So the goal was to make sure that we weren't getting spam entries. We think that there's a low risk of that. So we're trying to, again, like reduce barriers and just see what happens. So we'll keep an eye on it. If we start getting spam entries, then we may have to put a password back into it. But I suspect we'll be fine. So you'll go through your name. If you are doing this as part of your job, too, we're interested in your affiliation, or if you're part of like a lake association or some other group, put that information in. It'll also ask some information about where you were monitoring, the depths you were at, information about like the net that you're using. And there's a place to add photos as well. So if you took photos and you have something where you're either unsure of or you think it's a spiny water flea, you can upload those photos there, and I get an alert when new reports come in. So I always go and check. And if someone has marked that they think they found something, then we'll also start working our way back to connect with you, and photos will go to Don and Val, and we'll connect with DNR as needed as well for those. The newest piece to the reporting app is this map. So when you enter now, the page looks like this. This is a live updating map, so it's continuously refreshing. So as folks enter new reports or have new monitoring activities, you can see where that's happening. So if you're someone who's doing monitoring at a number of lakes, it can be a way to make sure you're not duplicating efforts. So you saw someone was just there, maybe you want to prioritize a different lake. And it's color coded. So the lighter colored markers are ones that happened in this monitoring season, and the darker colored markers are from previous monitoring seasons. If you go right now, it looks just like it does on the screen. The light markers are there. Those are all of our test runs. So those are about to get deleted out, and it will soon only have dark markers, but we wanted you to at least be able to see what that might look like. So you'll see the map, and that can give you some information as well. And then if you jump over to the submissions form, then that's where you'll enter in all the information for your monitoring activities. And I do want to note, it's super helpful when you are able to log your monitoring. It gives some great data for Don and Val in terms of search efforts. Even if those are zeros, it's really important data to have. But also, it's really important data that we can have when we're trying to share about the various work that we're doing, both across Extension and the Research Center. Being able to show kind of like the efforts that are happening as a result of these programs are things that funders like to hear, and other groups. So it's helpful for us to have that information too for multiple reasons. Last year, we had 15 different folks who participated. So if you were one of them, thank you. And we had 44 different adventures that we saw people do, and 32 different lakes. and no new detections of spiny water flea. So that's our results from last year. That's again been some growth from years before. So we're really hoping to see more of that growth as well. Okay, I'm going to turn over to Don for the frequently asked questions. Then we'll have a few minutes left for questions from you after that.
Okay, thanks, Megan. Okay, frequently asked questions. Can this be done from a canoe or kayak? Yes, absolutely. Depending upon your comfort level in those sorts of watercrafts, we've done some surveys in the boundary waters for spiny water flea, which is obviously canoe only, and we've taken a 0.5 meter diameter net. Based on your comfort level, you can certainly do this from those types of watercraft. You just have to exercise caution, of course. Will our zebra mussel veliger net work? Yes, it will definitely work. The spiny water flea are large. They'll be collected by any sort of plankton net that you have. Again, the only drawback is that if you're towing a really fine mesh net midsummer, even late summer, when phytoplankton, cyanobacteria, for example, can start to really become abundant in lakes as those lakes warm up, you're going to get a lot of debris in there that could challenge your effort to see all of the zooplankton in there as well. It's just going to be a little bit more effort to sort through that material. But if you have it, use it. I think that's perfectly fine. How do I find the deepest spots in a lake if I don't have a depth finder? Go to the middle. No. So typically, it's a good question. So the protocol that we recommend, right, is to go to the deep point and to do 3 successive vertical tows, and then to go to 2 more deep locations as some distance from that deep point and to repeat that. So in total, you will have gone to 3 locations. Hopefully one of those locations will be near or fairly near the deepest spot in the lake, and 2 other locations. And in each of those 3 locations, you'll do 3 successive tows. So that makes a total of 9 net tows in a lake. And the reason that we prescribe that is there's been research up in Canada a number of years ago to ask that question: how many tows does a researcher, a volunteer, anyone out there have to take in order to ensure detection if spiny water flea are actually there? And that's good. Now, if it's weather permitting or whatever it is, if you can only get out to a deep location and tow it once or twice or 3 times or 2 locations, the point is, if you can't do that full suite of 3 tows at each of 3 locations, that's okay. If the time and commitment that you can make is more limited than that, any data is great data. Okay, it doesn't have to be the full complement of 3 tows at each of 3 locations. Okay, what kind of weather conditions are best for spiny surveillance? So calm days and evenings and nights are best, simply because the boat's not going to push you, the wind's not going to push you around, and you'll be able to get your net close to the bottom, right, within 1 meter or 1.5 meters of the bottom, and that will ensure that you're sweeping it through as much of that water column as possible. As noted, spiny water flea tend to go deep during the day, and you could be sampling all day long in the surface, or if it's windy, you just can't get that net down there. So calmer conditions, and it's just nicer to be out there in calm conditions as well. And then, oh yeah, so our 3 plankton tows at 3 sites really necessary? I guess I covered that. I didn't see that bullet coming up. That would be the ideal, but if you can do any portion of that, that's fantastic. So do what you can. We've encouraged to not sample too close to shore. There may be some instances in which spiny water flea inhabit nearshore areas of lakes, but if you're in the weeds, if you're in those weed beds near shore, chances are, if spiny water flea are in the lake, they're not hanging out there. They tend to be more of an offshore, open water zooplankton, where they can retreat down to darker areas to avoid fish during the day, and that's why they tend to be out there. So if you can be away from shore 100 yards or so, that would be ideal. Okay.
Great.
Yep.
Okay, so I see we have a few questions. I remembered one last point I was going to make, and that was the other new thing that we have today is through evaluation that we've done, we've seen that one of the biggest barriers is the cost of the net to folks participating. And MAISRC was kind enough to throw some money at this program for us to try to get a loaner system set up. So we have the first couple purchased. We're going to do some spring trial runs and make sure the system for checking those out works well. And then in time for the monitoring season, we should have a broader suite. The goal is to have 10 that are spread fairly well across the state, both in kind of the area where we already know spiny water flea to be present, as well as in areas where we'd love to see some extra monitoring happening. I'm actually bringing a kit up to Brainerd this weekend, so there will be one right in that area that Don mentioned already. So watch for those as well. Again, like there will be 10 of them available. There's no fee to rent them. You'll just have to sign a release and make sure you return it. So we'll have details that'll come out by email on that. If you are signed up for this webinar, that means that you're on the mailing list to get notifications for the program as the season moves forward. So watch for more updates about that that loaner kit system. The kits will have just about everything you need. The things they won't have is a boat, or I think it doesn't have flashlights or things that require batteries. You'd want to bring your flashlight, and it won't have like alcohol or paper in it. So those would be the things you need to provide. But we even have little jars in them. So if you do find something, there is a jar in it ready for you to use. So those are the loaner kits. And I see we have some questions coming into chat. So I think I'm going to stop screen share and we can start working through some of the questions here. I see there's one asking about the specifics of the net. So we have a form on the website that we've popped up a few times. It was on the screen right before I shut it off, but that's the Spiny surveillance site that has the specifics, and it does have, I think, a little bit different size than the one that Don was using in the video. It's a 30 centimeter diameter, and I think that's the standard that comes from that company that Meg found with that 500 micron mesh. So that should be fine, unless you have anything to add to that, Val or Don.
No, I was reading that one. It looks fine.
Great. Yeah, and I think those look a bit different too. They have like the just the tube end versus having like that cod end that screws on and off too. So both
They're
are
a
okay.
little easier. It's a little easier to deal with. Sometimes that cod end gets sand in it, it gets stuck, you know, the one that like Don uses. And so I actually think the drain tube is a little bit easier. Just make sure you've got that drain tube really clamped shut before you lower your net. There's nothing like pulling it up and realizing it was open and, oh, guess you're doing it again.
Yeah.
There was another question too about whether or not the spinies are going to get like caught in the mesh down toward the bottom. They'll actually get caught in the mesh anywhere in the net. And so that's the trick. And Don shows in some of those videos more about how to rinse down the net. You dip it once and then you use your squirt bottle or whatever you've got to rinse down the net. And then you can look in there and see if you see the little black eye spots. So he goes through all that in the training videos. But we can guarantee you that those spines do catch on the mesh, and you just have to look for them.
Yeah.
Let's see, another question, Don, this one's for you. How
Yeah.
close do you want to get the net to the bottom?
I would try to stay, a lot of these stay a meter off the bottom, maybe a meter and a half or so off the bottom. It only takes one experience letting that net into a mucky bottom and bringing it up and realizing that you've brought up a bunch of the bottom. And it takes a little while to rinse that off. It's kind of discouraging, but if you have a marked rope or if you have a depth finder and you can actually watch your net going down and you can position it to within a meter and a half of the bottom, you should be okay. But once the hoop hits the bottom, and it drags a little bit, you're going to create a plume, and then you're going to collect all that in your net when you come up. So I just try to stay off the bottom a meter and a half, and I think you'll be fine.
So the next one, ice out has been happening sooner the last few years. Will that make them become active sooner? And I was wondering if you also, Don, know a water temperature that people could be watching for to say, oh, now is the time to start.
Yeah, those are all really good questions. The phenology, the seasonality, the timing of that is all going to start to change. Excuse me. The reason it takes a while to actually detect them is the populations have to grow from the bank of eggs in the sediment, and that's going to take a few rounds of reproduction to populate the population, to increase the numbers of individuals in the population high enough to see them. The other thing is that spiny water flea, they eat the other zooplankton, which themselves emerge from the sediments. And so in some ways that their presence is delayed by a few weeks behind things like Daphnia and some of these other native zooplankton that they feed on. So it's the timing of this and all the factors involved are sort of too complex to have a single water temperature. That would be a good timing or start point for beginning to look for spiny water flea. So the best I can say is lakes around here in the Duluth area start to reveal spiny water flea in late June, early July. It might be a little bit later along the Gunflint, might be a little bit earlier in Lake Mille Lacs. Of course, it depends on lake size as well. I saw there was one comment about what lake spiny water flea prefer. They tend to prefer deeper, less productive lakes, so lakes that we call like oligotrophic or mesotrophic. So very green water lakes, shallow, warm green water lakes are not the ideal habitat for spiny water flea at all. And if you look around at the lakes that have been invaded, it's probably not a big surprise that lakes along the Gunflint Trail tend to be deeper, colder, less productive populations that are thriving. So we'll see. There might be some barriers, thermal and productivity barriers to their expansion eventually throughout the state. But then there was another question there about how to split effort, and I'm not sure what to tell you. I going to two lakes with partial effort, I think could be more valuable than to go to one lake with if you're going to sort of split time that way. I think that would be interesting to do that. If there's a thriving population there in that lake, chances are less than 9 net toes, 2, 3, 4 net toes is probably going to detect spiny water flea in that system. But again, I just want to say just a couple things. One, zeros are really important. Knowing that something's not there is like it's as valuable as knowing it is there, because everything that we're trying to do is know really that something isn't. I mean, that's good medicine to know that it's not there. If it is there, it's interesting. It's really it's exciting from the position of an aquatic invasive scientist, researcher, and it will mobilize a lot of interest in that and effort in that. And so obviously we want to know if there's new hits, but knowing that there aren't animals there is really important. And that, if you live on a lake or near a lake, you can go out a couple times in a year, once in July, maybe once in August and September. That's great as well, because of the way in which these populations go up and down, right? I mean, you go out and you walleye fish, and sometimes you catch and sometimes you don't, right? It's kind of with spiny water flea too, right? Their populations might be susceptible based on density or position in the water column or not.
So this one might be for me. What are the guys that dart all over in the white bucket? If they're about a ½ inch long, and maybe slightly greenish tint, I suspect you may be catching amphipods. Just about everything, lots of things can dart, so that's a little tough for me, but the ones that really zoom, if you look at them close, you'll see that they kind of look like they're laying on their side, and they're kind of a little bit of almost a C shape, and all their legs seem to be on one side, and they're called side swimmers. They're amphipods. So those are, and they can just shoot around in your sample. Whereas the zooplankton and copepods and things, other types of zooplankton tend to swim in kind of bursts of speed. Amphipods really have some good speed on them. So that may be what you're seeing. I'd have to see a picture. So if you can get them to hold still enough, and you can take a picture, I'd be happy to help identify those. Yeah, 50-degree water temperature, you're gonna have zooplankton, that's for sure.
Yeah, so Kevin, yeah, thanks for pointing that out. So a lot of zooplankton, like spiny water flea and daphne and others, overwinter in the sediments and then emerge and grow. But there's a whole other group of zooplankton called copepods that tend to overwinter in the free swimming stage. And so there's zooplankton ever present out in a lake, under the ice, in the open water. But this group that spiny water flea belongs to, and their primary food, those tend to be much more seasonal in terms of their presence in the water column. So as lakes warm up, the density of zooplankton and the diversity that you see will continue to escalate.
And you can tell a copepod, it almost looks like a little tiny torpedo.
Yeah,
And this is a little torpedo shape with a tail, and then the females will have two little egg sacs hanging off the end of their torpedo shape by their tail. And so they're very good food because they're active all winter. They're good food for the fish. So that's the plus side of that. And I think Christina asking about a sample form, so we have on the website, where were you in the lake, what depth, how many toes, blah, blah, blah. So I think people are wondering if we have something simple they could maybe print out and take with them so they remember to write that all down so that when they get back, they have it. And we haven't, because we asked for so few things, we haven't thought of that, but that's something that we could easily put together if people wanted to do that. I mean, just a reminder that paper kind of disintegrates if it gets wet. So, so you do have to be careful with that.
Yeah, Kevin noted there's just Meg told me to buy, and then there was details on a net. Yeah, that's a great net. Yep, 500 micron, 30 centimeters, totally fine. Yep, go for it. That'd be a great one to buy. Yeah. I mean, half meter diameter is kind of ideal. It's just going to collect more water, but 30 centimeters, it's going to be more easy to manage, especially if you're in a smaller watercraft. You're not going to be reaching out as far, dragging it into you. Obviously, when you do this work in a canoe, which I've done, it's good to have some sort of a rain jacket on. I mean, you are going to get wet, right? You're going to have Nitex mesh all over yourself, trying to stay stable and still. Wear your life jackets.
And the other thing is, the smaller Nitex is really expensive. And so the smaller nets will be a little cheaper, a little more affordable.
Good point.
And I'm pretty sure that's why Meg was recommending those, besides the fact that it's a common net size that this company we're recommending is making. Are there other questions that folks have? And I see we've run over time. Oops.
Yep, that's just fine. If folks have questions, feel free to shoot us an email. You can reach me at the AIS Detectors at umn.edu email, and I'm happy to pass anything on to Don and Val that I'm not able to answer myself too. So if you have more questions about like biology or the nitty gritty of the monitoring, we can get them looped in. And yeah, if you find stuff and you're unsure, again, feel free to take a picture and snap it. There's the option to put a maybe in a did you find a spiny water flea? So and we'll send those in. Don and Val have been great at looking at those when I send them over. So thanks so much. And we will have this posted to YouTube, I think was the other piece. So you can watch her recording there too if there's any piece you want to revisit. We'll get it up there. It usually takes a week or so, maybe a little bit longer because we're about to host a conference. So watch her in a couple weeks, and we'll send out the notice when it's available on YouTube for training, and then you'll be getting messages from us with reminders about when it's good monitoring season and things like that. So thanks for joining us, and thanks again, Don and Val, for joining and sharing everything.
Absolutely.
Yep.
Thanks, Megan. Thanks, Val. Thanks to everybody that was here and for all your volunteer efforts if you participate this summer.
Yep. And remember, zeros are data. Zeros are real data. We need those. So thanks, everybody.
Thank you. Bye-bye.